Thank you, Pramila, for your kind introduction. Thank you for inviting me here to present my work. So today I'm going to talk about a subpopulation of microglia, which is HOXB8 subpopulation of microglia, and I'm going to show you the data how HOXB8 microglia functions in the brain, how a genetic mutation in HOXB8 microglia leads to very specific behavioral output that resembles autistic behaviors and OCD-specific behaviors, and how it communicates with the neuronal circuit. So repetitive behaviors are evolutionarily conserved behaviors. It's found in invertebrates like Aplysia, C. elegans, and in more vertebrate species like mouse as well as non-human primates. And in children with autism, repetitive behaviors is manifested as hand flipping behavior or repetitive lining of the objects, which later during the development of the child leads to speech regression as well as social isolation. And approximately 1 in 44 children suffer from autism and autism spectrum disorder. The numbers could be much less with the new statistics available. And if you look at the repetitive behaviors observed in adults, it could be manifested as obsessions as well as repetitive thought patterns, which could be also triggered by fear or anxiety levels and panic attacks. And approximately 1.2% of the population suffer from OCD, specific behavior, and nearly 6.8 million people suffer from fear or anxiety. This number also has elevated quite significantly with the COVID-19 pandemic. So here is a human case, my own child, Dave. He's a high-functional autistic, and he exhibits quite a lot of autistic behaviors, such as repetitive verbalization, repetitive visual stimuli, repetitive task looping. He has sensory issues, he has social interaction deficit, and social isolation deficit, and he has problem with math as well as comprehension and cognitive coherence. So recognizing his problems way early in the development, we have him undergo several therapies like speech, OT, social therapy, and ABA therapy, which had profound effect in the way how he is right now. It had quite impact on your language, W questions, his attention, his self-regulation, his self-balance, as well as self-balance as well as his overall development as an individual. Where he's now able to go to school and he's able to sit with his normal neurotypical kids, and he's continuing his education. Okay, so if you look at the symptoms and comorbidities of repetitive behaviors, we find cognitive comorbidities, behavioral comorbidities, we have medical comorbidities, several biomarkers has been discussed here in the meeting, as well as work from several labs around the world. And there are also several genetic elements or genes that are highly responsible for the behavior. But what you see in the Venn diagram is the common core symptoms of the disorder, which includes impaired social communication, social interaction, social reciprocity, also compounded with the restrictive repetitive behavior and anxiety, increased anxiety levels. So as Pramila has pointed earlier and many times, that there are only 2 FDA-approved drugs currently available for autism, and there are several factors that contributes to the development of ASD behaviors, and some of the factors that are contributing are pointed here. That includes environmental toxicants, maternal immune activation, microbiome, microbiota, metabolic pathways, neuromodulation, maternal stress, non-genetic and epigenetic changes, as well as genetic components. So, and many of these factors have been touched by the talks that we already have heard. So our responsibility just not is restricted to our research, but we have, as a parent, quite a huge number of responsibilities as to what would be the educational, education that the child need to have. What are the therapies that we need to carry out all throughout the life of the autistic child? What are the careers that are available for them? How do we deal with comorbid features that they exhibit throughout the development of their childhood, entering into teenager and adolescence, as well as adulthood, and what are the medical care available? And besides all these things, how do we self-manage ourselves, as well as how family can manage the whole concept of autism and autistic child, caring and parenting. So, my interest in this particular work starts from understanding a very simple form of behavior, repetitive behavior, and I started searching for, a gene that could specifically act on a cell type, which, if we identify which cell types are present in a particular neural circuit, and if we have a behavior that's connected to that particular neural circuit phenotype, then we possibly be able to understand this behavior at a very fundamental level. So my search led to this gene called HOXB8 gene that was discovered in Dr. Kapeke's lab in 2002. The genetic mutation in HOXB8 to stop the expression of the HOXB8 gene led to a very specific behavior, which was grooming behavior that was excessive in the HOXB8 mutants. And the cell type, surprisingly, turned out to be microglia, which is a non-neuronal cell. And the neural circuit that on which this microglia was acting turned out to be the corticostriatal circuit, which I discovered later in 2017 after entering in Mario's lab. So my question was, what is the causal link between HOXB8 microglia and the repetitive behavior? So, as pointed, the loss of HOXB8 gene function leads to very specific excessive grooming behavior, which is a repetitive behavior that leads to hair loss in male mice and hair loss followed by pathological lesions in female mice. And since the HOXB8 gene precursor or HOXB8 microglia precursors comes from the bone marrow, they thought that bone marrow transplantation could be a way to identify whether one could rescue the phenotype. And in this experiment, they took bone marrow from the healthy mice and they transplanted that to mutant, and they did the reverse experiment. So interestingly, what they found was the animal that received the bone marrow from the healthy controls started showing the grooming behavior that was more wild-type-like level, implying that there is a rescue phenotype. And the animals that received the bone marrow from the mutant animals, they started developing the hair loss pathology as well as the lesions on the body. So in either direction of research, it showed that bone marrow transplantation was rescuing the phenotype. However, the bone marrow transplantation rescue was only observed in 60% of the animals that were tested. So this was shown earlier that HOXB8 lineage labels very specifically only microglia in the brain, and that was done using lineage analysis, where HOXB8 Cree driver was crossed to a ROSA YFP reporter, and that gave rise to only microglia, and no other cells in the brain was labeled with this particular HOXB8 lineage cells. And here is the bottom panel that shows the entry of the HOXB8 microglia starting from postnatal day 2 all the way up to postnatal day 90. So now it's very well accepted that microglia are involved in multiple diseases, not only in repetitive behaviors related to OCD or autism, but also in schizophrenia, Alzheimer's, depression, multiple sclerosis, and ALS as well. So it's very important to understand at the fundamental level how these microglia functions in the brain. And my question was, how does HOXB8 microglia communicates with the neural circuit and how it controls the behavioral changes as we found in the HOXB8 mutant animals. And why neural circuit? Because neural circuit process information via synapses, and synapses, as we all understand, are the fundamental unit of the brain. So my thought was, if we understand the neural circuit and synapses more carefully, and if we understand the impact of HOXB8 microglia on the neural circuit as well as synapses, we might be able to understand the complex behavior that is mediated by HOXB8 microglia neural interaction. So to address that, I used miniature endofluorescence imaging technique that was developed by Mark Schnitzer at Stanford University, where it's a miniaturized form of microscope. The microscope is as small as your nail finger, and it weighs approximately 2 grams. A mouse can hold this microscope on the top of its head and walk around in awake state, and we can induce very specific behavior of our interest to see what happens to cells of interest inside the brain. And shown here is a freely behaving mouse, which is given a social stimuli, and they were able to show that the activity inside the neurons mediated by the calcium change was found in different parts of the hippocampal neuronal cells. So based on this technology, we thought, why don't we try to measure calcium inside HOXB8 microglia? And to do that, the same technique was used and same miniscope was used as previously established. So I expressed GCAM5, which is a calcium sensor, very specifically in HOXB8 microglia by using Krelok's recombination. And I implanted a lens, which is 4 millimeter in length, inside striatum, which is also part of the corticostriatal circuit that controls the repetitive behavior, and performed a very simple experiment of spraying a little bit of water on the face of the mouse. So a healthy mouse that's moving around and normally behaving now enters into grooming behavioral state in response to a small water spray. And as soon as the mouse enters into the grooming behavioral state, we wanted to find out what happens to microglia in the brain, especially HOXB8 microglia. And interestingly, what I found was that in each of the individual microglia that are shown on the left-hand side, we were able to capture very small calcium transients, very different from what we have seen with the neuronal calcium transient. But nevertheless, even though it's small, it can be captured, and we can actually quantify and we can measure the DF over F, which is change in the fluorescence from the ground state to the increased calcium fluorescence state. So this individual traces corresponds to calcium transient coming from individual HOXB8 microglia in the brain when the animal shifts its behavior from normal behavior to the grooming behavior. So the heat map shows you what happens during the pre-grooming behavioral state, where you see a flat line, and then as soon as the animal enters into the grooming state, you see an increase in the calcium within HOXB8 microglia. And then it comes down once the grooming behavior stops. So, and that was reproduced in multiple mice, and I was able to show that in fact, we can induce these calcium transient very reproducibly, not only between subjects but also within sessions, within multiple sessions of the individual subject. Shown here are all the calcium transient coming from individual microglia in response to shift of its behavior from normal behavioral state to the grooming behavioral state. So here is a comprehensive heat map corresponding to 12 individual microglia showing increased fluorescence of calcium in response to grooming behavior and decrease in the calcium fluorescence once the behavior stops. So to find out whether this is not artifact, but this is a real signal coming from microglia, I also implanted the lens in a second brain region, it's in medial prefrontal cortex, which is also part of the corticostriatal circuit, and showed that, in fact, even in medial prefrontal cortex, in response to the grooming behavior induction, you're able to get calcium transients. And in this case, I used GCAM6F, which is a much faster calcium sensor compared to GCAM5. So using a different chemical sensor, calcium sensor, using a different brain region, we were able to show that calcium transients are present in response to the induction of the grooming behavior. And this was again reproduced in multiple sessions. I also showed that if you have multiple grooming bouts, you can get multiple calcium transients. So calcium transient was somewhat directly proportional to the number of grooming bouts that the animal was engaged with. So, because the water stimuli induced the grooming behavior only for a short time interval, I developed another, technique where I wanted to see if I can sustain the grooming behavior for a longer time. So, in order to achieve that, I used a mineral oil drop stimuli, which is a small drop of oil on the surface of the skin, which irritates the mouse skin, and the animal engages itself in grooming behavior. And once the animal is engaged in the grooming behavior, which is more consistent compared to the water spray, we were able to show that we can, generate calcium transient in Hoxby8 microglia. And what you would see is that different microglia exhibit different number of calcium transient. Some are silent and some are overactive. And one of the reasons why these variations might occur with respect to microglia could be the fact, how close are these microglia are in relation to the neural circuit that are being active in response to the behavior. And you can classify these transients into different transient subtypes based on the shape of the calcium transient. So if you look at the session-wise comparison of individual microglia, you can see that there is differentiation between how an individual microglia would show its behavior in terms of session 1 versus session 2 versus session 3 versus session 4. So it's variable, but it's still there. So you can reproduce the same calcium signal measurement over and over again in the same location of the brain by recording the cells from the same location as well as same animal, and demonstrate that in response to the induction of the grooming behavior, you can elicit the calcium transients. So here are some quantifications showing you the cellular response of the active Hoxby8 microglia transients, and and also the correlation plot that shows all the microglia are simultaneously active in response to the induction of the grooming behavior. So here is a slide that shows you the histological verification of the cells that I am actually measuring. And these are all the microglia that is being seen through the lens as and when the animal is engaged in the grooming behavior. So to again verify whether whatever we have seen with respect to corticostriatal circuit is true, I also did control experiments of stimulating viscous and measuring the calcium responses within basal cortex using the same approach. In basal cortex, we did observe the oscillations, but the calcium transient were not similar to what we detected with the frontal cortex as well as the striatum. So the next question was, what is the nature of calcium transient in Hoxby8 mutants? Because that was more interesting, because this mouse has excessive grooming behavior and it just keeps on grooming. Now we wanted to find out what is going on with that particular population of microglia. So to address that, we created a genetic mutation in Hoxby8 gene in the exon 1 and expressed the GCAM6F within Hoxby8 mutant microglia and performed the same experiment. So we expected that we would see a lot more calcium transients in response to the genetic mutation that we had created. But to our surprise, we were not able to see any calcium transient. Everything was flatline in these subjects. So when we looked into what's going on in individual cells, they were having calcium, but the calcium was not being regulated. And that's what is shown in this video, where you see these cells are all there, but the calcium will not go up or down, but it remains exactly like the way how it is, for any number of days, for any number of months. So this is just one video showing you the calcium signal in awake behaving mice. And this is after 30 minutes, and you see the calcium levels, remains exactly in the same way. So it's a super saturation level that these microglia exhibit their calcium levels, and that regulating capacity of calcium is lost in these mutant microglia. So what I showed you so far is that there is a problem with the Hoxby8 mutant microglia, and now I wanted to find out, what's the correlation between the Hoxby8 microglia to the neuronal activity as well as repetitive behaviors. And in order to address that, I used optoelectrophysiology as well as optogenetics methods. And optoelectrophysiology is where you can take a brain slice and you can stimulate the HOXB8 microglia optogenetically, and then you can record electrical activity from an adjacent neuron. And what I found was that in pre-stimulation condition compared to post-stimulation condition, that the continuous blue light stimulation resulted in enhanced spike frequency as well as instantaneous frequency, as well as the, but it reduced the event interval, the spike interval. So it suggested that the neuronal activity is altered in response to HOXB8 microglia stimulation. And shown here is another proof that light stimulation is activating the neuronal cells. So if we use CFOS, which is an early gene expression marker, we can see that CFOS is increased in response to light stimulation only in the implanted hemisphere, but not in the spared hemisphere, which means only the cells that are seeing the light being stimulated on HOXB8 microglia will show the CFOS increase, but on the other hemisphere, which does not have an implant, you don't get the CFOS expression increase. So this again gave us one more level of proof that CFOS, which is an early gene expression marker, is also increasing in response to opto-stimulation of HOXB8 microglia. So this was restricted to only HOXB8 microglia, but when we looked at all microglia, we weren't able to see the CFOS increase. So that was also very interesting. Why only HOXB8 microglia and why not all microglia? So I showed in a molecular psychiatry paper that if you optogenetically stimulate HOXB8 microglia, you can induce grooming behavior even in a control mouse. And that is shown in the second and the third figure, where you see the increased grooming time as well as increased grooming bouts. However, if you do the same experiments in C57 black 6 controls, you don't get such kind of increase in grooming behavior. So the optogenetic stimulation induced grooming behavior was restricted only to HOXB8 microglia. When we performed all microglia by using IBA1 Cree driver, which labels all microglia, we were able to see no behaviors were induced, be it grooming behavior or be it freezing or be it anxiety-like behavior. So that was quite puzzling and interesting in its way. So what I showed you was that HOXB8 microglia is involved in inducing the neuronal activity, but HOXB8 microglia is also controlling the repetitive behaviors. And now neuronal activity increase is what is connecting the repetitive behaviors to the HOXB8 microglia stimulation. So one question was, what happens when HOXB8 microglia are actually stimulated? What's going on the surface of the HOXB8 microglia, or how HOXB8 microglia is reading the neural activity patterns around them? So in order to perform that, in order to address that question, I performed the optogenetics of the HOXB8 microglia to see what I can observe on the surface of the microglia membrane. And I used Kv1.3, which was already shown earlier, to increase its expression in activated state. And surprisingly, in response to optogenetic stimulation, I was able to detect increase in Kv1.3 expression on surface of the HOXB8 microglia in medial prefrontal cortex as well as dorsomedial striatum. However, in control experiments where our control hemisphere where we did not have any implantation, we weren't able to see any Kv1.3 expression. So in control experiments, there was where there is no behavioral induction, you also won't get increased expression of Kv1.3. So that suggests that Kv1.3 expression increase is in relation to the induction of the grooming behavior. So more recent experiment has shown that if you continuously stimulate HOXB8 microglia for 2 weeks, by giving multiple stimulation per day, ranging between 2 minutes to 7 minutes, you can induce pathological hair loss behavior even in a control mouse. So that's a non-implanted mouse. The pre-stimulation looks like that, and post-stimulation will show increased hair loss. But interestingly, unlike a disease model where there is a continuous stimulation of microglia and continuous repetitive behavior, we can recover the hair loss behavior once you stop the stimulation. And shown here are the quantification that shows there is increased phase 3 as well as phase 4 grooming in response to the light stimulation of HOXB8 microglia. So I showed you that optogenetically stimulating HOXB8 microglia actually increases the neuronal activity pattern, which also leads to repetitive behavior. I showed you the HOXB8 microglia increases the Kv1.3 expression on microglia, and we also detected the higher levels of calcium in the HOXB8 mutant microglia that probably is responsible for restrictive repetitive behavior. And now with RNA sequencing analysis, we have detected one particular calcium channel, which is Cav2.1, to be differentially altered in HOXB8 mutant cells compared to the wild type cells. And I'm currently conducting the experiment to see if we can observe any changes in the Cav2.1 in HOXB8 mutant microglia versus control microglia. I don't have all the data here, but what I can show you is Cav2.1 is expressed in HOXB8 microglia, and there are 4 different types of Cav2.1 can be observed in brain slices. We can see HOXB8 microglia which are not in contact with Cav2.1, which are in contact with Cav2.1. We can observe some of the HOXB8 microglia that are enwrapping the Cav2.1 as well as engulfing Cav2.1. And if you look at what happens in the non-HOXB8 microglia, we see more or less the same. It would be interesting to see what happens in the mutant microglia. So what is the implication of Cav2.1 altered expression in mutants? We can test that hypothesis by directly manipulating at the genetic level the Cav2.1 on HOXB8 microglia. And secondly, we can also use pharmacological interventions to see if we can reduce the signaling mediated by Cav2.1 to see whether we can alter the repetitive behavior. So, I showed you that HOXB8 microglia genetic mutation or optogenetics leads to the repetitive behavior. And towards the last part of my slide, what we have identified is that the two population of microglia works in tandem with each other. One serves as a brake and the other serves as an accelerator. So by perturbing either through genetic manipulation or by perturbing through optogenetic manipulation of HOXB8 microglia, we have induced repetitive grooming behavior as well as anxiety-like behavior. And this is conducted based on the changes in the calcium levels of the HOXB8 microglia. So summarizing what I showed you, HOXB8 microglia elicits calcium transient in response to grooming behavioral induction. Repetitive grooming behavior increases neuronal activity within medial prefrontal cortex. Repetitive grooming behavior increases the expression of Kv1.3. And HOXB8 mutants show calcium regulation defects in HOXB8 microglia. So with that, I would like to acknowledge Dr. Kapeke's lab as well as Dr. Mario, who has allowed me to conduct all these experiments, all the funding agencies that supported my work, the Brain Foundation that has invited me to come here and give my talk, and thank you very much for your attention.